Good morning or good afternoon, as the case may be. Hey, what's up, guys, and welcome to Friday. Hope you have had an awesome week. We're going to go ahead and spend today talking about how the stuff that we learned in the last class, meiosis, rolls right into genetic inheritance and Mendelian genetics. And you're probably like, oh my gosh, I'm just getting down meiosis. I totally get it. Meiosis and mitosis are both complex processes that require a lot of thought. So I'm going to start off today by reviewing some of the stuff that we've been over just to give it a little bit of context and to go ahead and roll it straight into Mendel. So let's get started. Last class, we talked about DNA and RNA being nucleic acids, which is one of the four types of macromolecules in existence that make up your body. DNA and RNA both act as information storage devices. Essentially, they're going to contain all of the instructions for making the proteins, which make you. So all the DNA and RNA are basically manuals to make you. And we learned that DNA and RNA, when a cell is ready to reproduce and make more cells, has got to be duplicated, and a complete set of instructions or chromosomes needs to make it into each one of the new cells. We learned that this happens through two major processes, mitosis and meiosis, and let's go ahead and review those really quickly as well. Okay, so we said that each cell is going to go through something called interphase, which is like most of the cell's life before it reproduces. It's childhood, adolescence, and whatnot. Interphase is marked by three stages, G1, S, and G2, where the cell grows in both G1 and G2, and it replicates or doubles its genetic instructions or DNA during the S phase, so it can go ahead and pass those on. Here you go, kids. I made this for you. So reproduction and division is achieved at the end of interphase in a cell by mitosis or meiosis. Remember, we started with mitosis and said that it went through four stages, prophase, metaphase, anaphase, telophase, or PMAT, which created two identical what we call daughter cells. And now if you're confused about that, daughter cells does not necessarily refer to the sex of the cells. In fact, most of the cells created by mitosis are not sexual cells at all. They are somatic or body cells. Daughter cells just means a cell that is the product of division. So we create two genetically identical daughter cells in mitosis, and this is great for our somatic body cells that need to reduce their surface area to volume ratio, or that need to repair, divide, or repair their DNA using the checkpoints in the cell cycle. But we don't always want to reproduce that way. Sometimes we want to reproduce offspring, and that has to happen through a process called meiosis. So contracting it with meiosis is going to happen in precursor cells to produce sperm and egg, otherwise known as gametes. It is going to create four of these cells. They will be haploid in their number of of chromosomes, which means that they're going to have one of each chromosome as opposed to two in diploid cells. And so they're going to have 23 chromosomes in humans because a normal diploid human cell has 46 chromosomes. And it'll go through 8 stages, 2 rounds of PMAT. So PMAT twice essentially without replicating the DNA in between to get that half number of chromosomes. So meiosis, why did we need it to be a haploid number? Well, we want it to be a haploid number because if we're going to have two different gametes combined to make one new body cell of the developing zygote or embryo, we're going to need to make sure that that zygote or embryo has the correct number of chromosomes. Every species has a different number of chromosomes that have to be present in its body cells, and in humans, that number has got to be 46. What happens if we have more or less than 46? Well, that's where we get into genetic disorders, such as Down syndrome or Turner syndrome, and it can affect the development of the child. So we went 46 chromosomes. If we used mitosis, well, mitosis is the asexual divisionary process that creates two identical diploid cells. If we use mitosis to make sperm and egg, then each of the sperm and egg would have 46 chromosomes, add those together, and the new developing zygote would have 92 chromosomes. I can tell you that zygote would probably never even get close to developing because that is a super improper chromosome count to build a human. If meiosis occurs, however, sperm and egg are able to have 23 chromosomes, and when they combine in fertilization to make the zygote, they make a new body cell of a newly developing organism that has 46 chromosomes, which is the proper number for human beings. Now again, each species is going to have their own number of chromosomes, and their sex cells will always have half the number of chromosomes that their body cells have. So meiosis achieves this haploid division by taking a diploid precursor cell, undergoing one round of PMAT to go ahead and separate homologous chromosomes. During this time, it also ensures that there is some genetic variety in the cells that it's creating, unlike in mitosis. And it goes over the process of crossing over, which is where homologous chromosomes swap genetic information, and also goes over the law of independent assortment, meaning that it's randomized as to which one of the original chromosomes goes into which developing sex cell. We then have PMAT again to go ahead and again demonstrate the law of independent assortment. It's randomized as to which one of the four new gametes is going to receive which chromatid. It separates those sister chromatids, which again we said were those duplicated copies of the chromosomes made during the S phase, and it creates four unique gametes or sex cells, each of which is not only haploid with 23 chromosomes each, but also has genetic diversity. So each one is genetically distinct from the other. Okay, so again, why can't we use mitosis? Because it creates genetically identical cells, and the two big things that we need for meiosis in order to have genetically diverse offspring that can survive a changing environment is crossing over and the law of independent assortment. I'd like to go very quickly into crossing over and the law of independent assortment again, just to make sure that you guys understand what's happening. During prophase 1 of our meiotic or meiosis division, we are going to end up getting our homologous chromosomes swapping genetic information. This swap of genetic information is going to make it so that each one of them has slightly different allele combinations than they had before. Remember, alleles are different versions of genes, so that means that the proteins that are going to be produced by this final swapped chromatid with this weird little segment of DNA on the end is going to be very different than this chromatid, which has none of that DNA segment. The same is going to be true over here. This chromatid that has the swapped end is going to make slightly different proteins than the chromosome without, therefore creating some genetic diversity in both the traits and the proteins that are being made in the offspring. Again, this happens during prophase 1 of meiosis. What is the law of independent assortment and also the law of segregation, which I don't think that I talked about last time because, well, it's a lot all at once. But the law of segregation and independent assortment is beautifully shown in this graphic. Here we have meiosis, and meiosis is producing the four different gametes. These are two different, what I say types. These are two, this is meiosis happening twice. So this is the cell undergoing meiosis over here with the law of segregation, and this is that same cell undergoing the meiosis over here with the law of independent assortment. So don't think of this as all being the same meiosis. I just wanted to show you that it's the cell undergoing it with this over here and the cell undergoing it with this over here. Hopefully that makes sense. Sorry for describing that in such a terrible way. My brain does not always work the best. Okay, so let's go ahead and look at the law of independent assortment. That's where we started with. So the law of independent assortment is where we say that the genes in non-homologous chromosomes are going to assort independently during gamete formation. What does that mean? Okay, well that means if we look over here, non-homologous chromosomes are going to contain different genes. So in other words, the R chromosomes, even though they have different cases of letter in front of them, this is a capital R and this is a lowercase, those are both going to count as homologous chromosomes because they represent the R chromosome, regardless of whether it's capital or lowercase. The same is true of the Y. The Ys represent the Y chromosome, even though one might be capital, one might be lowercase. Now, when we go ahead and we sort these, when we actually go through the division process, notice that in each one of the cells created by that division, it will have one copy of the R, whichever one, and one copy of the Y. The other one also has one copy of the R and one copy of the Y. Now, what is true is that they can either inherit them like you see here, right, where you see this has the little r and the big Y and the big R and the little y, or they could have inherited it so that this one had the little r and the little y, and this one had the big R and the big Y. So it doesn't matter which one they go into, and that helps to randomize which one of them ends up in what combination in your gametes. And that also, in turn, helps randomize what the kid ends up being. Okay, the law of segregation is similar. It just says that the two alleles for each gene separate during gamete formation. In other words, what it really just means is that we don't have both R's together in the same one of these cells. So notice you'll never see an RR or a YY in any of these gametes. And that also helps to make sure that not only are we getting the full number of chromosomes that we need, but we're also making sure we're getting a diverse shuffling of the alleles for those genes. So those are the ways in which meiosis promotes genetic diversity, which again is important so that our offspring can exhibit variation in traits and not be exactly like the parents. Why? Because if the environment changes and the environment selects against the traits the parents had, well, maybe the kids have a greater chance of escaping that environmental pressure if they have slightly different traits than the parents. So that's why we want that variation. Okay, wow. So that was a lot to go over. That's sort of just covering everything we've gone through in meiosis and mitosis so far. Now, how does this apply to genetics? Okay, so way back in the day, hundreds of years ago, there was this dude named Gregor Mendel who was an Austrian monk. And Gregor Mendel was really curious about how children inherited the traits of their parents, but not quite exactly like the parents. So he was wondering, like, so why is it that some kids really favor one parent over the other? And why is it that some kids don't look very much like their parents at all, assuming that they haven't been adopted, you know? So this was a big mystery back then, and people were thinking that if you could crack the problem of inheritance, which is what they called this, then you could go a long way in figuring out how life works. And that was really important to a lot of people. So Gregor Mendel got together with the abbot of his monastery, and he was like, dude, I'd like to do some scientific experiments. And the abbot was like, dude, totally, man, like that's super awesome. Dude, go at it, yo, like figure out God's creation. And Gregor Mendel was like, awesome, man. I just want to like breed some mice up in my room. And the abbot was like, whoa, hang on. I'm so sorry, but having mice have sex in your room is going to tempt you sinfully. And Gregor was like, what? And the abbot was like, yeah, I'm so sorry, but I just don't feel comfortable with you being so close to animals having sex. And Gregor was like, dude, I'm a monk, and I'm kind of chose this life. And the abbot's like, my word is final. Don't question my genius. And so Gregor Mendel was like, okay, well, could I maybe do this experiment using pea plants? And the abbot is like, of course, pea plants don't have sex. And Gregor Mendel was like, actually, you're right. They don't. I'll just use pea plants because they don't have sex. Pea plants definitely have sex. But Gregor Mendel didn't push the issue. He wanted to do his experiment, and voila, Gregor Mendel got to do his experiment. And so what he did was he bred pea plants together until all the kids had the exact same traits as the parents. When he saw a purple blooming pea plant, he bred it with another purple blooming pea plant so that all the pea plants ended up being purple blooming. And then he did the same for pea plants for white blooms, and then he made a different line of pea plants that had yellow seeds versus green seeds, and inflated seeds versus wrinkled seeds, and all these other things. And he made all these very independent lines. Now, why was he doing this? He was doing this because Gregor Mendel had this really interesting idea. He thought that there was some kind of molecule or substance inside of these pea plants that was being passed on. And he thought if he could sort of make it so that he knew exactly what type of substance it was. So, for instance, in the purple pea plants, it would just be the purple pea plant substance versus the white pea plants would be the white pea plant substance. And then if he could sort of breed them together, if he saw a pattern, he could figure out exactly how these traits were being passed on. Now, that might be a confusing way to say it, but basically he was just trying to make sure that he knew what traits that these plants normally would have, so that then when he bred them together with plants with other traits, he could try to see whether or not there was a pattern as to which traits were inherited by the offspring. Maybe that's a better way to say it. Okay, so what was the result? So the results of Mendel's experiments were kind of incredible. So when he took a true breeding, in other words, one of those pure green pod plants he had bred, and one of the true breeding yellow pods, when he combined them together, all the kids had green pods. no matter what he did. When he take a true breeding green to a true breeding yellow, all of the kids had green. Now that is a really weird result, okay? And he did this over and over and over again, and it just kept proving true that only green pods were coming out of this breeding cycle. And he's like, what is going on with this? Like, come on, man. And so he figured that there had to be some version of the substance for green that was somehow overriding or more powerful than the version of the substance for yellow, and that each time that you bred a green together with the yellow, the green substance would just overpower the yellow and create yellow offspring. Now, Mendel was on the right track. Of course, Mendel didn't know the genetic stuff that we do now, so it was impossible for him to be able to say, oh, this is due to the fact that certain alleles are more powerful or dominant than others. But we do know. So let's go ahead and take a look at the molecular stuff that's going on to see if we can understand what's happening to Mendel's cross. We're going to do that with a monohybrid cross. Now, the first thing to understand before we go any further is that the substance that Mendel was working with was, of course, DNA. And that DNA contains something called genes. Okay, genes are segments of DNA, so just short little links of the genetic material that code for a protein, or well, I'll just say proteins. So essentially, it just means a little bit of DNA that codes for a protein. What would that look like? For example, if I went ahead and said TAC, let's see, TAC, CCG, and CTA, or actually CAT, we'll say. So these are all, notice these are all bases from DNA, T, A, C, and G. Okay, let's say that that particular sequence would equal a protein that makes your eyes blue, or something like that. Now, I'm not saying that it does, but I'm just saying that it could. Okay, so it'd make a protein that makes your eyes blue. Whereas another version or allele of that gene, okay, so an example allele maybe would be TAC, CGC, CAT. And just that little change might mean that the protein that makes your eye color will make your eyes brown. Whoa, brown. Okay, so you can kind of see how just small changes, and I'll go ahead and type this, small changes in your in your genes can cause big changes in your traits. Okay, and this is a really important concept. So really what Mendel was looking at was he was realizing that certain alleles were more powerful than others. Certain versions of genes were more powerful than others, and they were overriding them. So really what Mendel was seeing was that the green P alleles were overriding the yellow P alleles. Okay, now how does this actually work? Okay, this leads us to the concept of dominant and recessive, which you guys have probably heard of before. So dominant and recessive alleles, what does that mean? Okay, a dominant allele is going to be a version of a gene that when present, okay, in other words, when it's possessed by an organism, always shows the dominant phenotype or trait. So if you guys remember, the genotype is what genes you have, and the phenotype is what you look like. We talked about this during evolution. Natural selection acts on phenotypes, which are the traits organisms have or their characteristics. So the dominant allele, when you have that gene, you will always show the dominant version of that phenotype or that trait. Okay, for the recessive alleles, it is a version of a gene that only shows the recessive phenotype when the dominant allele is not present. Okay, so in other words, you have to have only recessive alleles for a particular trait in order to show that phenotype. All right, so what do we actually mean when we're talking about dominant and recessive, and why is it that you can have more than one allele for a particular trait? Well, actually, this should make a lot of sense when you combine it with what we learned previously. So previously, we were talking about how chromosomes come in pairs. For instance, this would be chromosome pair 1. You would get one homologous chromosome from dad and one homologous chromosome from mom. We said that homologous chromosomes were going to have the same genes in the same order. So here, homologous chromosomes equals same genes in same order, but different alleles for those same genes. Okay, so in other words, you inherit your mom's unique way of making chromosome 1 stuff happen, and you inherit your dad's unique way of making chromosomes 1 stuff happen. Now, this is really important because it means that for every gene that you express in your body, you have 2 copies that you're actually using to make proteins in your body. One copy is dad's copy of that allele, and the other one is mom's. So you have 2 alleles that 2 alleles expressing every one of your traits. Okay, that is a big thing to say. Okay, and that means that you could receive multiple potential combinations from your mom and your dad depending on what they have. So let's go ahead and kind of dive in to see what that would look like. Okay, so whoops, back again to the concept of us talking about chromosome pair number 1, just because it's easy to talk about. We've already kind of discussed it. So here we have our homologous pair of chromosomes at chromosome pair 1. Okay, and let's say that for a particular allele, dad has the dominant phenotype. Now, or excuse me, the dominant gene or allele. So the dominant allele is always shown by a capital letter, and the recessive allele is always shown by a lowercase letter. Okay, and so if we are looking at this, then that means that if the dad has the copy of the dominant allele, we'll say that the allele's letter is A, it doesn't really matter, and mom has the recessive copy of the allele. Actually, no, I'll go with this. And mom also has the dominant copy of the allele. Then you would receive 2 dominant copies, and this would equal what we call homozygous dominant. Okay, which means that it'll express the dominant phenotype because only the dominant allele is present. Now, homozygous, what does that mean? So homo means the same, and zygous just has to do with the zygote or the diploid cell that you're talking about. So really, just think about zygote as meaning gene. So it means to have 2 of the same allele or same gene. Okay, so homozygous dominant, and the dominant refers to the fact that you have 2 uppercase letters. So homozygous dominant, and you will express the dominant phenotype. So say this is eye color, let's just pretend for chromosome number 1, you will express the dominant eye color, whatever that eye color is. Okay, now, we're going to go on and go to a different scenario. So let's say that instead of a dominant allele, your mom had the lowercase or recessive allele. What would happen? Okay, so if you had the capital A lowercase a, this would mean that you had the heterozygous condition. Hetero means different, and if we say zygous means gene or allele, it means you have 2 different ones, the dominant and the recessive, but you're still going to show the dominant phenotype because remember that the dominant allele is always shown when present. So it doesn't matter if you have 1 copy or 2. If you have even 1 copy, it's going to show the dominant phenotype. So big A, little a, heterozygous is still going to show the dominant phenotype. It's only when you get up here to, let's say the dad had the recessive phenotype as well, it's only when you get to that point that you have a homozygous recessive. Remember, homozygous means same allele, so you have 2 of the same alleles, but they're recessive, and you get the recessive phenotype or the recessive eye color. Okay, and this is just talking about one particular trait. Okay, there are many, many, many traits that we talk about, but this is just one. Now, what does that mean? Holy cow, well, that means that if we go back to Gregor's experiment from before, and we assume that green pea pods are dominant and that yellow pea pods are recessive, and we go ahead and we cross them together. So we imagine we go ahead and take a homozygous green individual and we multiply it by a homozygous yellow individual, which I'm going to represent by using the lowercase form of the same letter. Then we would go ahead and get something that looks like this. So this is called a Punnett square, and it is a tool for calculating the inheritance of traits within offspring based on their parents. So if I go ahead and do this, I'm going to put the first individual on the top, the green one. So here's the green pea pod plant. And then over here, here's the yellow pea pod plant. And all I'm going to do is I'm going to go ahead and I'm going to put the letters, distribute the letters into the boxes here to go ahead and show what the genotypes of the offspring are going to be. So here I'm going to put capital G little g, capital G little g, capital G little g, and capital G little g. So I went ahead and distributed all of my letters. And if you notice, big G little g is the heterozygote condition. And because the heterozygote condition has a copy of the dominant allele, it's going to show the dominant phenotype. So that's why they were all green. Whoa! Okay, so that should make a whole lot of sense. So all of those are green. And that was amazing to Mendel. Now, Mendel went ahead and he performed a second cross. So this was the first cross that he did, and he went ahead and he performed a second cross called the monohybrid cross. See here, we're getting back to that. Okay, so the monohybrid cross. And the monohybrid cross was when he went ahead and he took two hybrids, okay? So he took the GG times the GG, so two heterozygotes here, big G, little g, and he crossed them together. And they were called hybrids because, we would call them hybrids because they have one big and one little allele. Wow, my pen is doing something weird here. Okay, so we go ahead and we do the Punnett square cross, and up here we put the parents, one letter per column, so big G, little g, and whoops, whoa, and big G. Oh, this is so frustrating. Little g. Oop. And then we go ahead and we put them all into the box, and we notice something interesting happen to the inheritance pattern as we distribute these letters. We end up getting a very interesting pattern. If you were to look at the ratio of dominant to recessive phenotypes, then you would end up getting 3-to-1 dominant to recessive, because all three of these guys have at least one copy of the dominant allele. So they're going to show the dominant phenotype. This one only has the recessive. They will show the recessive phenotype. This ratio right here came to be known as the golden ratio of Mendelian genetics because it happened over and over and over again in his experiments, thus showing that he was correct in thinking that, hey, there's something special about the pattern of inheritance of traits. So that is the introduction to Mendelian genetics and what is meant by recessive and dominant genotypes and phenotypes. Now, if you're still wondering what a genotype is going to be this right here. So the genotype would be the letters or alleles that you actually have. So this would be known as its genotype. Okay, but the phenotype of this individual, what they would look like based on those letters, okay, would be, in this case, green would be their phenotype because it is based on their genotype. Okay, so phenotype is what you look like, genotype are the letters that you receive. And this leads us to that monohybrid cross. Now, the monohybrid cross was Mendel's first little breakthrough, but he soon discovered that traits weren't just, you couldn't just look at one trait like the color of the pea pod or something. That was a bit simplistic, wasn't it? Because plants have way more traits than just one. And so he was able to extend this to the dihybrid cross. And the dihybrid cross is going to be what we're going to take a look at next class. We'll take a look at what ratios he was able to get out of the dihybrid variety and see if we can't figure out how that would affect inheritance patterns. And then we're going to take a look at non-Mendelian genetics. So we're going to take a look at what happens when you are facing a situation that doesn't quite fit to Mendel's, what might be causing that. But I think we've gone on long enough for this. That's all for me for today. So have a great time, guys. I hope this made sense. Email me with any questions or concerns you might have, and I'll do my very best to address those in a timely manner. Stay safe and stay well. Can't wait to talk to you guys again soon. All right, take care.